Hi everyone, my name is Daniil Gataulin. I'm one of the Senior Research Analysts covering biotech here at Chardon, where our vision is to help identify companies with high potential for investment returns based on creating real value for society through disruptive innovation. It is my pleasure to introduce Paul Bresge from Ray Therapeutics to the second series of our virtual ophthalmology summit, where the focus will be on retinitis pigmentosa and other inherited retinal diseases. Paul, welcome.
Thank you so much for inviting me.
All right, so let's jump right into it. If maybe start fairly broadly, if you could provide a brief overview of Ray Therapeutics, maybe touch on the company history and highlight some of the programs.
Sure, of course. Firstly, thank you for highlighting, retinitis pigmentosa. It's very personal to me. Just a little bit of personal background. I got back into the biotech space in 2011, and that was shortly after my middle daughter, her name is Tamar, shortly after she was diagnosed with retinitis pigmentosa. And that led me to co-found a company called JSITE, which is a retinal progenitor cell therapy program targeting retinitis pigmentosa. And I took that program through very early proof of concept up to what is now a partnership in a phase 3 program with Santen Pharmaceuticals. Very important work that the field is doing for this very high unmet need. In terms of Ray, it's an optogenetic gene therapy company for blinding diseases. The difference between the company that I worked with before with JSITE, it's primarily targeting patients in the earlier stage of disease to stop progression of the disease. What we're doing at Ray Therapeutics with our optogenetic approach is really targeting patients at the late stage of disease. And unfortunately for these patients, because a lot of the work that's being done in the field with cell therapy, with gene therapy, very important work, but it's really targeting to slow down progression of disease. What we're actually looking to do is restore vision in these patients. The company was formed in 2021. There was a seed round that was led by 4Bio Capital. And then we were very fortunate to receive a $4 million CIRM grant. That's California Institute for Regenerative Medicine. The seed round was $6 million. Lead indication is retinitis pigmentosa, followed by Stargardt's disease. And then we do have a follow-on program in geographic atrophy as well. Last May, so this is May 2023, we announced a $100 million Series A round led by Novo Holdings.
Got it. Thank you for that overview, and thank you for sharing your personal story as well. You mentioned that Ray utilizes the optogenetic approach. For those who are fairly new to this space or new to this modality, can you explain how optogenetic approach works and why do you believe it's the right one to treat retinitis pigmentosa and potentially other inherited diseases?
Sure. I mean, I'm talking about, our program specifically. We're using essentially the most light-sensitive protein that is found in nature. And then we have highly bioengineered that protein specifically for human vision restoration. We're using AAV gene therapy to deliver that protein to the retinal ganglion cells to give them similar function to what the photoreceptor cells are responsible for. Again, as I said earlier, there's not much hope for these patients at the end stage of disease. So optogenetics is a very exciting modality for these patients. The other thing that's important in the context of retinitis pigmentosa in particular is that it's a disease with many different mutations, and the optogenetic approach is independent of the actual causative gene. So it provides, potential treatment for a really broad patient population.
Got it. And so there have been several optogenetic programs in development from several companies. Can you talk about what makes your program unique, what sets it apart from others, and maybe even tell your thinking in terms of what gives you confidence in your program's success?
Sure. I mean, I'm very grateful to the other companies that have had some very exciting programs and continue to have exciting programs in the optogenetic space. The thing that we have seen is safety, which is of course the most important thing, and then I would say very confidently proof of concept. When we look at GenSight, for example. They've had already published, really remarkable results, for patients using optogenetics. The programs to date, and GenSight again is a great example, and Jose Sahel, is just a remarkable, human being who has done so much for patients with blinding diseases. In that program, the patients are required to use goggles because the protein itself is not light sensitive enough. So the goggles provide additional light. As we know, patients at the end stage of disease will, do anything to have improved vision. And that's why the program is, of course, very exciting. Unfortunately, I believe at least that there are, potential limitations when you think about the regulatory and commercial pathway for combining a device with a therapeutic. So what we have done at Ray, again, as I mentioned earlier, is taken the most light-sensitive protein, and then we're really the first company, to move towards clinic with a highly bioengineered optogenetic protein. So it's been engineered for all of the complexities of human vision restoration, and that's why we think that it'll be very game-changing in the field, which is already a very established and exciting field for patients.
Got it. So I wanted to dive in a little bit deeper on a couple of comments that you just made. Number one is the use of goggles. And as you mentioned, some of the earlier programs utilize proteins that are not light sensitive enough, and they do rely on goggles for light amplification. So both from the user perspective and from the regulatory standpoint, how limiting are goggles?
Yeah, it's a great question. So I think so from the regulatory standpoint, I think it's combining again a device with a therapeutic is challenging. From the patient's perspective, the goggles require a lot of training. If I'm correct, I think it's up to about a year of training. The other issue that I understand, is a limitation with the goggles is that because they have so much light, they become very hot, and the patients cannot use them for very extended periods of time. So that, of course, is not ideal. The difference here is that we expect this protein to be sensitive in ambient light. So it's about 1,000 times more light sensitive than the original program that went into clinic with optogenetics. That was the RetroSense program. So again, given the fact that it is so much more light sensitive, the fact that it has been bioengineered really specifically for human vision restoration, and the fact that, we believe that patients will not be required to use the goggles for the limitations that we've just discussed. Again, we think that it's a huge differentiator in the field.
Got it. And in terms of training, so you mentioned that goggles do require extended training protocols, in some cases maybe up to a year, for patients to understand or to be able to interpret the signals that they perceive. What about your approach?
Yeah, no, we don't expect that patients will require training. The interesting thing, or one of the really exciting things about the program, we expect to see efficacy very quickly within a couple of months of dosing patients. When we think about traditional, retina trials, because again, most of these trials are looking to slow down the progression of disease. There's a typically, at least a 12-month, waiting time, or 18-month to endpoint, in order to show any significant slowing down of rate of disease or improvement. We expect to see patients improve within, as I mentioned, within a couple of months, and we do not expect any additional training. for their vision.
Got it. Okay, that makes sense. And so the vector that you use for your RTX-015 program, that's the lead program in RP, as well as in Stargardt. So the vector is AV.7M8, which is licensed from Adverum. How did that partnership come about, and why did you choose that vector?
We chose the vector, and we were very fortunate to enter into an agreement with Adverum on the basis that that is the best vector in our opinion for transducing the retinal ganglion cells. So that is our target cell in RTX-015, again for RP and for Stargardt's. It made most sense for us to license that from Adverum.
Got it. And in terms of the route of administration, how is RTX-015 administered?
It's an intravitreal. It's a very simple, in-office procedure.
Okay. And since that vector is being used in wet AMD trials, and Adverum is testing seroprophylactic regimens to reduce the risk of inflammation, do you plan on using similar prophylactic regimens?
Yes, so we do plan on using a prophylactic regimen, and we have engaged some of the top retina KOLs in the country, actually even beyond the US, who've reviewed our protocol and agree that it's very appropriate. And of course we're paying attention to to potential inflammation and obviously going with a very strong regimen.
Got it. Makes sense. I wanted to spend a few minutes to talk about the results that you saw in your preclinical trials. So if you could maybe spend a few minutes and highlight some of those, specifically which models did you use, what efficacy results you saw, and how specifically you assessed for efficacy in preclinical models.
Of course. I still remember the day that Sean Ainsworth, he's our chairman, and he was actually the founder of RetroSense, again, the first program to go into optogenetics. I still remember the day that he approached me and showed me the data that came out of Dr. Pan's lab, and I was just absolutely blown away. As you can imagine, over the many years that I've spent now in the space and seen every animal model, and obviously because I have such a personal interest for my own child, I follow everything. I pay attention very, very closely. I was just again absolutely blown away by the data, but not only the data, which I'm happy to share. It was also produced in what I consider really the highest bar animal model, and that's the triple knockout. So these mice are born blind, completely from birth. So they haven't even developed the cortical pathways, because of that. And they also mimic the patient population very nicely because they have no functioning photoreceptors. In those mice, Dr. Pan was able to show about 50% of visual acuity restoration and near normal contrast sensitivity. Because these animals don't have the developed cortical pathways, we expect even better results in humans that have had vision previously. And again, the translatability of that model mimicking the patient population, we have very, very high expectations. So that was absolutely game-changing for me, and that was what got me so excited about becoming involved in co-founding the company.
Got it. And very interesting. And how specifically do you assess for vision restoration in these models?
So it's the optomotor test, which is a very standardized, well-known test, a very objective test. Dr. Pan, he's very thorough. The assays are extremely robust. We've actually been able to replicate that data under the umbrella of Ray in now three independent studies, again under Ray's umbrella. So we're very confident in Dr. Pan's data set.
Got it. Makes sense. And what do you see as potential challenges translating those results into humans with RP?
I think we've considered what those challenges are. Have a very experienced management team. Have been involved in ocular gene therapy programs for decades now. And I think we've addressed them. We've done now a couple of NHP studies. As I mentioned, several studies under the umbrella of Ray and in mice. We're just very excited to get to clinic and and get into patients essentially.
Got it. And I guess one more question regarding preclinical results. How long did it take to see the results in those preclinical models?
So I believe that Dr. Pan's studies were like 1 to 2 months, and and the ones that were again conducted under the Ray umbrella. I mean, of course, with mice, because they can't tell us what they're seeing, they're tested at different time points. We looked at the data, over the life of the studies, and we have data that goes out, as long as 12 months, and shows that it's the efficacy is sustained. But again, when as it translates to humans, we are expecting, very quick efficacy, relatively, particularly with other programs within about a couple of months.
Got it. And in terms of safety signals, can you comment on what you saw there and which doses did you get to?
So I can't comment on the dose and the mouse, doses specifically, but yeah, I mean, very, very well tolerated and very safe as well. And as mentioned, we previously completed an NHP study, and we have another in-life study, right now, NHP.
Okay. Yeah. Okay. Got it. Makes sense. And I guess that brings us to my next question. In terms of other gating items, so you just mentioned one ongoing study, but what else is there for you to do before you can file the IND for 015?
Yeah, we're just finalizing all of the standard IND enabling studies and preparing for the IND. We plan to file the IND early next year, and obviously don't expect any delays.
Got it. Okay, makes sense. And in terms of so there are 2 programs for 015, RP and Stargardt. Are they on roughly the same schedule?
No, we plan to initiate the Stargardt's trial about 2 quarters after we initiate the RP study. So that will, hopefully both studies will, initiate, next year again with the RP study, in the first or second quarter, and then the Stargardt study to follow about 2 quarters after.
Got it. Makes sense. So for the potential study design, can you walk us through what you're expecting it to look like? Maybe also talk about the patient population and potentially some of the signals, efficacy signals, safety signals that you'll be specifically looking for.
Sure. So the study design is really a typical rare disease study design to establish of course safety and therapeutic doses and efficacy. One of the great things about the study is that our safety population is also our treatable population. So we do expect to see efficacy in even in the really early patients. In terms of patient population, these patients will be legally blind, essentially, all the way to completely blind, like no, even potentially no light perception. And we are looking for validated endpoints. So the MLMT, the mobility maze, and visual acuity. And we will of course be looking at other endpoints that are really well known to the agency.
Okay, makes sense. So in terms of the drug itself, you are working with this third-party manufacturer. You have a partnership with Forge. Can you talk about that partnership and how you're making sure that you're not going to face any CMC issues or any other issues that many other companies in the space have faced recently?
Yeah, it's again a great question and a really important one. Being in the space for so long, we've learned those lessons. And actually, very shortly after forming the company, I think at that time it was myself and my then COO of the company. Literally, the very first thing that we embarked on was a CDMO search. So we put together a group of advisors and consultants to help us, and we did a landscape assessment. And I have to say that Forge came out as the best potential partner for us. And I am so pleased to be working with them. They have delivered above and beyond all expectations. They work very, very closely with our team. It really is like a true partnership. And so far everything has gone phenomenally well on the CMC side. We have a lot of expertise on the CMC front, and again, we're paying very close attention. I'm well aware that CMC can be a bottleneck, and we are, I think, very well positioned, in our CMC program.
Got it. That's good to hear. All right, we're nearing the end of our session, but I have a couple of more comments. So as you mentioned in the beginning, sure. You completed a $100 million Series A few months ago. Wanted to ask if you could comment on the lead investors, and that's A, and B, which milestones do you expect this raise to carry you through?
Sure. So, as mentioned, Novo was our lead investor, and I have to tell you that when we embarked on our outreach for the Series A, they were the very first company at the top of my list, and I met with them. Actually, it was the day after Labor Day, so I identified them as as my dream lead. So I was very pleased, when that came to fruition. And then we have other just outstanding, and really supportive groups in the round. We have Deerfield, we have Norwest, we have Platinus, MRLV, so the venture arm of Merck, and for Bio Capital. And I mentioned Norwest as well, right? And we again, we were just so grateful that the round came together, and it was important for us to do a $100 million raise. Getting to your second question, and that was because we wanted to make sure that we were funded to a very important inflection point, and there's enough funding for us to get all the way through to BLA in our lead program. So we feel like we're in a very fortunate position.
Got it. Okay, and I guess I'll end the session on the last question. For investors looking to enter the space, what do you think some of the investors are missing about Ray Therapeutics?
I mean, again, I'm going to use that word grateful. I'm very grateful that I don't think the investors are missing anything, just based on the last round and the fact that it was so heavily oversubscribed. I think that the investors really validated what I saw in the program and what our team sees in the program. So I feel fortunate that I don't think they missed anything. I hope that the investor community continues to follow our program. We have very high expectations for filling a very high unmet need. There's an absolute focus on the patients here, and if we're able to deliver a therapy to the patients, of course the investors will do very well.
Got it. Excellent. Thank you very much, Paul. I really appreciate you taking the time to join us for the summit, and look forward to catching up with you again soon.
Okay, thank you so much. It was great talking to you.